A team led by Dr. Robert Tranquillo at Minnesota University set out to develop a new and improved method for bioengineered tissue grafts. They'd be readily available, off-the-shelf grafts that possess the characteristics and properties of native tissues. Previous grafts were prone to infection and rejection by the host due to the presence of foreign living cells, while the new method involves the removal of living tissues to produce an acellular graft, which has little to no chance of bodily rejection. The most important aspect of these new grafts is their ability to grow and develop with surrounding tissues as individuals age. Traditional grafts, despite being accepted by the body, must be continuously replaced at different stages of development, making them inefficient. The newly designed grafts are superior to previous methods due to their ability to integrate into the surrounding tissues. They assume the natural shape and function of the host tissues, show acceptance by the host immune cells, and show proper development and growth alongside the host tissues. Native blood vessels contain large amounts of connective tissues such as fibrin and elastin to ensure that they maintain their shape and function under high pressure. This is precisely why the scientists constructed the original scaffolding out of unique cells that produce fibrin called fibroblasts. Fibrin is a protein used mainly in blood clotting, which under normal bodily conditions would be replaced with a collagenous network. The replacement of fibrin by collagen is precisely what the scientists desired, as the collagen graft provided a suitable structure for host cell integration. Major issue with traditional grafts was that they contained living cells and therefore had the possibility of being rejected by the body. This is due to the fact that foreign antibodies entering the host cause a rapid immune response where they are targeted and quickly destroyed by host immune cells. This harmful response had to be resolved, as it proved to be a major roadblock in the progression of bioengineering technology. They resolved this issue by using a specialized solution that removed all living cells from the synthesized grafts, making them acellular and therefore removing their ability to cause a harmful immune response. This allowed for universal implantation, eliminating the need for individual and personalized grafts. The largest issue concerning traditional methods was their inability to grow and develop alongside the host, meaning that they had to be continuously replaced by newer and larger ones to accommodate growth. This process was severely cost inefficient, as multiple grafts had to be synthesized and multiple surgeries performed just to provide treatment for one individual. The creation of an acellular graft provided a solution for bodily rejection, while the use of collagen and scaffold or synthetic tubing provided an environment suitable for host integration. These grafts were biodegradable, therefore once they were completely taken over by the host cell, they dissolved, leaving behind grafted tissues with equal or greater strength than native tissues. Using these changes in methods, the scientists synthesized and implanted pulmonary artery grafts into 3 two-month-old lambs. They monitored their functions over time and recorded their experimental results. They found that the grafts were growing at a normal rate with the lambs and that the lamb's own cells, including smooth muscle, had integrated and began to secrete elastin, giving natural properties to the grafts. This gave the grafts the same mechanical properties of native arteries, allowing them to grow and assume natural function. The newly developed grafts offer many promising future possibilities, as 1 in 1,000 children are born with congenital heart problems, requiring multiple surgeries throughout their childhood. These new grafts could possibly reduce the number of surgeries and expenses, sparing children from multiple surgeries and families from severe debt. This new method turns an acellular, non-living graft into living, functioning tissue universal to all individuals. Another benefit of the new method is that the grafts can be grown, stored, and used in patients in need without the extensive process of creating personalized grafts. Overall, the new grafts save time, money, and distress caused by multiple surgeries.